In this video, we will continue our discussion about melanin. So, in this slide or in this video, we will discuss about the genome-wide CRISPR screening of melanogenesis. So, this is the basic structure of the experiment. That is, they took the Cas9 MNT1 cell lines, and in these cell lines, Cas9 is induced when they treat them with doxycycline. So right now they have not induced any, they have not treated these cells with doxycycline. So what they did is they induced, infect these cells with the different guide RNAs with the CRISPR library infection, different guide RNAs, and after that they did the puromycin selections to get rid of the cells that are not infected, and then they treat the cells with doxycycline, and after treatment of the cells, the after treatment of the cells with doxycycline, what happens is that the genome editing will take place, and then they separated the hypopigmented cells and the uh, hyperpigmented cells using flow cytometers, that is flow sorters, and that is shown here. The hyperpigmented cells will have low side scatter, and the hyperpigmented cells will have high side scatter. This is the basic overview of the experiment. So in the panel B, they have shown, they have separated the cells with the in the least 10% of the side scatter as the low side scatter flow sorter, and the high 10% of the side scatter as a high as the high side as the hyperpigmented cells. And from visualization we can find out that we can see that the low side scatter will, and the high side scatter cells have high pigmentation. Then they did some bioinformatics using this CAS-TLE confidence score, and in this case they can find out some of the genes that are responsible for melanogenesis or melanin, that are responsible for melanogenesis, and some of these genes that they have identified are also previously reported. So this indicates that their bioinformatics analysis and their approach is more or less correct. However, they have not worked on finding out, they have not worked too much on this high side scatter, as they uh, hypothesize that this high side scatter might be because of other reasons such as more granularity of the cells. So their analysis is mostly focused on this low side scatter. Later on, they did this CAS-TLE confidence score, and in this CAS-TLE confidence score, they can see that, and these confidence score are based on two biological replicates. That is, they uh, treat the cells with CRISPR guide RNA library infections on two different at two different times. Uh, so, and also this cell sorting will also happen at two times. So these are the results for the these are the results of the average of the two biological replicates, and the results that they have discussed represented here are of the false discovery rate less than 10%, and they have also seen some genes that are related to melanogenesis in the false discovery rate less than 20%. So from that they made a point that the false discovery rate of less than 10% is a more conservative approach, and they have a confidence level of 95%. So in this they have highlighted the genes that are already discussed in, that are already been reported in the literature, which are responsible for melanogenesis as a red, and the the black genes are the genes that are that are unidentified before them and are responsible for melanogenesis. So knockout of these genes leads to the low side scatter, or in other words low melanin formation, and then they combine these genes using their algorithm to find out whether they form a some kind of complex or they are related to specific categories or not. So in this figure, if the genes are, and the size of the bubbles uh, represent the proportion of this guide RNAs that are proportion of this genes that are responsible for melanin formation, and the with the attaching of this bubble indicates the formation of complex structure. uh, so and when these are not attached, it means that they are not forming any complex structure, but they are related to some similar family. That is, in this case they are related to this regulation of RNA splicing, and in other case, that is in ATP AP3 adapter, they form a complex. These genes combined together form a complex. So these are the genes that they are able to identify which get affected whenever there is a low melanin formations. So from this experiment, they are able to identify a whole lot of genes that are not be previously reported and are responsible for this melanin formation. If you need to understand this whole CRISPR screening studies in detail about this melanogenesis, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.